This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM This Week in Microbiology, episode 149, recorded on March 30th, 2017. This episode of TWiM is brought to you by Blue Apron. Blue Apron is the #1 fresh ingredient and recipe delivery service in the country. See what's on the menu this week and get your first 3 meals free with your first order and free shipping by going to blueapron.com/TWiM. Hi, everybody. I'm Vincent Racaniello, and you're listening to TWiM, the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Well, hello there. How are you?
I'm very well, thank you. Looks like we're finally in spring here in New York City. How about you? You're always in spring.
Well, I have something to tell you. Spring this year was incredible in Southern California because finally we broke 5 years of drought and had wonderful, gentle, fine rains. What I understand Native Americans call female rains, as opposed to precipitous male rains. Anyhow, went out to the desert, the town called Borrego Springs, in the Anza Borrego State Park, and I had never seen anything like it. We used to have a house there, so I know the place well. It was wall-to-wall flowers. Wow. And if you want to see some, go to Facebook. Look for Elio Schachter's Facebook site, and I have a bunch of pictures.
Oh, wonderful. I will do that. I was just going to say, did you take pictures? Great.
Absolutely.
All right. Also joining us today from Ann Arbor, Michigan, Michele Swanson.
Hello.
Welcome back.
Thank you.
We missed you.
I was going to ask Elio about the female rains and mushrooms.
Yeah.
Oh, this has been a great year. Mushrooms are all over, and I've been giving talks right and left about mushrooms. So,
that's great.
Great year all around.
Michele, last time on TWiM you weren't there, but Elio made this statement: mushrooms get you sick once.
No, you can have any mushroom once. You can eat any mushroom
That's
once.
it.
Yeah. I either was there or I listened to the episode, but I love that.
You can eat any mushroom once.
Once, yeah.
I love it. Also joining us from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
How have you been?
I've been well.
Michael's back home. He was such a trooper last time. He Skyped from LAX.
Yeah, that was an adventure.
Ouch.
Michele, he couldn't get back to the hotel in time, so he stayed at the airport and waited for TWiM. It's dedication. I love it.
Did you just go in the family restroom and lock the door?
Yeah.
I don't know if the Wi-Fi would get through the door, but it worked.
He went to the lounge, Michele.
I went to the American Airlines Admiral Club. And went in there, and they had decent Wi-Fi, and it all worked well.
Yeah, we appreciate it, Michael.
It was a great set of papers. Unfortunately, Michele, I didn't get to speak with the authors because we were in the wrong time zones.
Right.
No problem. We have a guest today, also from Ann Arbor, Michigan, the Department of Microbiology and Immunology, Pat Schloss. Welcome to TWiM.
Thank you. Glad to be with you all today.
We've mentioned your name many times on this show. We've done a couple of your papers, and it's good to have you. Now, you're in the same department as Michele, right?
I am. Actually, I don't think Michele likes hearing this all the time, but she was the search committee chair of the search that brought me to Michigan. So Michele and I have more than a connection of colleagues. I really appreciate her mentorship and leadership here in the department. So
I should get a % of your indirects, shouldn't I?
It comes out now, Pat.
Pre or post Trump?
Well, it's good. Well, indirects are good.
Oh, that's great. Well,
he's a wonderful colleague. He's proven our wisdom time and time again.
Thank you.
Pat, when did you arrive at Michigan there?
So it was September of 2009.
Wow.
So
that's your first job, right?
No, it was actually my second faculty position. I'd previously been at UMass Amherst for 3 years before coming here. So I really admired Michigan because if you remember what was going on in 2009, the economy, and especially the Michigan economy, was not doing so well. And so for the University of Michigan to double down in hiring faculty, I thought was amazing, and has continued to exemplify that and be just a great place to work, where there's been a lot of vision from the administration and other colleagues.
At the time, too, Pat was working on a model system, an insect model system to study microbiome biology in the gut. So, which ties in well with the paper we'll do later.
Oh, interesting.
Yeah.
And I know we've talked, Michele, you've talked in the past about how you received funding to make an anaerobic unit so Pat could do his work there. Is that right?
Yeah. Well, Pat can talk more about that. That's down in his wing of the department.
Yeah, the med school has been really helpful, and they initiated a project called the Fast Forward Initiative, and the microbiome group here received funds to set up a room where there's maybe 6 or 7 anaerobic chambers. Mm-hmm. We have sequencing capacity. We have other cultural capacity. So it's a great resource to have all those tools, and then also great colleagues who are really interested in microbiome research.
Yeah, we actually put a picture of that room on TWiM once a long time ago.
Yeah.
Yes. So they have microbial ecologists, they have clinicians, they have computational experts like Pat. Mm-hmm. It's really a great group.
Pat, where are you from originally?
I grew up in Columbia, Missouri.
Wow.
Yeah.
Columbia, Missouri. Now,
was your family associated with the university?
Yeah, I'm what I call an academic brat. So instead of being a military brat where you move from base to base, I moved from college town to college town. But I did junior high and high school in Columbia, and really grew to love the Midwest from my time there. And so it was funny, when I was moving from Massachusetts to Michigan, people were trying to tell me how East Coast Ann Arbor felt, and I was like, no, I want to go back to the Midwest. there are people that told me that Amherst was Midwest, and I was like, we're east of New York City, right? So,
where did you go to college, Pat?
I went to Cornell University in upstate New York.
How about that? So did I. Of course, I graduated probably 20 years earlier than you did. I graduated in 1974.
Yep, it was about 20 years. Yeah. So I got my undergrad in 1997, and then I stayed on for my PhD, which I defended in 2002 or so.
It's funny. I'm going there next week to do a seminar, and going to do a seminar and a podcast, and I haven't been there in a while. What did you major in? Biological sciences?
No, I actually got my degrees in biological and environmental engineering.
Okay.
And so, I loved math and I loved biology. And so to me, that seemed like a great combination. And
it certainly has served you well.
Yeah. So I was in Riley Robb Hall, which is right next to Wing Hall. Mm-hmm. And so I spent a lot of time in the basement of Wing Hall with people like Anthony Hay and Bill Giorsi and Gene Madsen and Steve Zinder. So I loved the microbiology faculty up there, who didn't treat me like an ugly duckling, but treated me like one of their own for hanging out and learning about microbiology, even though I was an engineer. So,
and what did you do your PhD in?
So I studied waste treatment and looked at the succession of bacteria in compost. And so people say, "How'd you get from compost to the gut?" And I tell them it's basically all
similar.
It's the one same easy thing. It's just a different reactor, right? So.
That's great. Did you also do a postdoc?
I did. I did my postdoc at Madison with Joe Handelsman.
Oh, right.
And I feel so fortunate that worked out, and it was just a great time of science, and learned so much and so much more than just science, and learning about teaching and about being a person and so much more. And certainly Joe's been a great contact to have over the last 15 years or so.
She's back in Madison now.
She is.
And that actually, if I may interject, I've been wanting to ask a question, and I think you're the right person, especially after your absolutely classic piece of work that you published with Joe Handelsman on the census of the prokaryotic world. Absolutely, you updated it recently. You have a new paper. So you did the census of the bacteria and archaea by their taxonomic affiliation. I don't know what to call that, by species, by whatever it is. You figured out how many there are. And I want to extend that to a different question. Do you have any idea how many more bacteria there are than archaea by weight, not by number of taxonomic units? Do you have any idea about that? Does anybody know? I assume there are more bacteria than...
The numbers that I've heard bandied about are about 10 to 1, but I don't. That's not a very hard number. That's pretty squishy. And
which way, Pat? 10.
So
times?
10 times the bacteria to archaea.
Okay.
With people doing shotgun metagenomics now, it should be easier to get at that, at least on a DNA basis, where we're sequencing all the DNA and we can see what's bacterial and what's archaeal. Mm-hmm. So I feel like I'm not the person right now to answer that, and I could have been better. I should have thought of that ahead of time, but I feel like it is. Like, of course, it's going to depend on the environment, but
right,
of course,
the bacteria certainly dominate. And on a weight basis, cellular weight basis, I'm lost there too. So,
well, this is good enough. Thank you very much. I appreciate that.
Pat, where did you get your interest in computational biology?
Yeah, so it actually goes back to Joe's lab. And so, of course, I was an engineer, and I loved taking differential equations and linear algebra, and wasn't quite sure how that was going to fit into biology and transitioning more towards microbiology. But so this was 2003, 2004, and I had sequenced a grand total of about 1,000 16S sequences from a single gram of soil. And so back in my day, that was a lot of data. Of course, today we can whip that out for not very much in a day or two. And so we had like this 1,000 16S sequences and didn't really know what to do with it. And one of the curves we can generate is a sampling curve, or what we call a refraction curve, where you plot on the x-axis the number of sequences you sample, and on the y-axis the number of taxa you've sampled. And that's a pretty simple thing, you might think. And so we were putting together a poster for NSF to present our research, and Joe said, "Hey, Pat, why don't you make one of those plots?" And I said, "That sounds like a great idea, Joe." And so about 3 years later, I finally made the plot because it wasn't so simple. But to illustrate to your listeners how valuable these tools are, that very simple procedure of clustering the sequences and then counting the number of types you have went onto a paper that was cited maybe 2,000 times. And it's, I wouldn't say it's my greatest work. It's maybe my most important work. But just a little bit of computational skills. I had taken one semester of programming as an undergrad in a language no one ever uses. But that helped me to learn the tools I needed to make that plot. And so people describe me as a computational person or a bioinformaticist, and I don't necessarily like that term because what I see is that I have a problem, right? So Joe says, "Hey, Pat, make me this curve." Or I have a problem where we want to identify the bacteria that are associated with colon cancer. And I look around and I don't see the tool I need, but because of my training and the training I give my trainees, we can do that. And so, okay, I'm a bioinformaticist maybe, but what we really try to do is put the emphasis on the biology and the questions that we're trying to get at. So that's, as a postdoc, is really where I started to develop those skills and apply them to our research.
So you would say you're a microbiologist, and when you have to solve your problems, you use computational approaches.
Yeah,
sometimes.
He's also being modest, though, because he's developed some tools that are now used worldwide.
For sure.
Right.
Yeah.
Mother, for example. I don't know if you could even estimate, Pat, how many times that has been used in papers.
Yeah, so I'm a little vain. So it's been cited about 4,600 times since it was originally published in 2009.
Wow.
Which just blows me away. Again, it's humbling to me because I don't feel like the methods are super sophisticated, but at the same time, they're really important and really valuable to people. And I love going to the microbe meeting or running into people when I go give talks who are undergrads or postdocs, and they said, because of your software, we were able to do this paper, and they light up, and I light up because that's, I don't know, it feels really good to know that you've made something that others find useful.
Absolutely.
it's transformational. It's up there with the Lowry protein assay, which used to be one of the most often cited papers when people used to do protein assays.
Well, thanks.
Well, and also lest people think that it's all done. There's plenty more to do, as we'll see when we get to your paper. And we are doing tomorrow on TWiM a paper which was just published where they looked at the blood virome of 8,000 people. And they say, we would have liked to translate all the open reading frames, but we don't have the computational power to do that and do any kind of significant blasting, So,
wow.
Right.
And I think, and you tell me if you agree, it's not just a matter of making more powerful computers, but better algorithms, right?
Right.
Absolutely.
And recruiting more people who can work at the interface of the biology and the computation.
Yeah, for sure.
So come join us, all you engineers.
I have to say, when I was a postdoc in the late 70s, so there were very few genome sequences. So we did the genome sequence of poliovirus, which was the first time that had been done. And it took us a year to do the sequence. And then once we had it, we couldn't do much with it except translate it, right? Because there was nothing to compare it to.
Yeah.
And back then you took your organism, you determined its sequence if you were lucky, and then you worked with it. You didn't think of doing more genome sequences because you couldn't compare them. You couldn't do anything with it. And that's what computational biology has changed. That it's brought people in the field who said, look at all this big data. Let's figure out ways to manipulate it. So it's really an amazing revolution. The American Society for Microbiology wants to tell you about their grant writing online course where you can learn the best practices in grant writing. Participate in this course to get an overview of the funding landscape. Learn what makes grants successful and get personalized feedback on the grant that you've been writing. This is a 7-part series taking place in the fall of 2017. Registration deadline is this summer, July 15th. You can learn more at bit.ly/ASMGWOC17. That's bit.ly/ASMGWOC17. Also want to let you know about the 18th annual Microbiology Student Symposium in Berkeley, California. I was a previous keynote speaker at the 2013 symposium, and I didn't actually give a keynote address. I did a TWiM, and you can check that out over at microbe.tv/TWiM. This year's keynote speakers are more traditional, but very good. The Queen of Quorum Sensing, Bonnie Bassler from Princeton University, and Edward DeLong from the University of Hawaii, who travels the world to map marine microbial communities in 4 dimensions. So if you're in the Berkeley area, you should check it out. It is happening, I think it's happening in May. More information and free registration, nature.berkeley.edu/calmicro. Berkeley is spelled Berkeley. nature.berkeley.edu/calmicro, CALMICRO. So we wanted to talk with you, Pat, today about a paper you just published in mSphere, and it's called OptiClust, an improved method for assigning amplicon-based sequence data to operational taxonomic units. And the first author is Sarah Westcott. And one of the reasons, we'll talk about the science, but first let's talk about the way this was submitted. This is called mSphere Direct. And you were the first person, not only the first person to have an mSphere paper when the journal first debuted, but now you are the first to have an mSphere Direct. So tell us about that.
Yeah, I guess I'm an early adopter when it comes to publishing. So I really have to kind of tip my hat also to another one of our colleagues, Mike Imperiale, who's the editor-in-chief of mSphere. To him and Stefano Bertuzzi and Tom Schenk, the current chair of the journals board, for coming up with this idea of mSphere Direct. And to step back a little bit for some of your listeners who might not be familiar with the more traditional peer review process, that when we want to publish a paper, we write the paper, and then we submit that to a journal. Some editor at the journal might get a hold of it, hopefully, and then they'll send it out to 2 or 3 reviewers. And then the reviews will come back, and then it's our job as an author to respond to those reviews and then resubmit it. And this might take 2 or 3 iterations, at the outset, the editor might say, this doesn't really fit our journal, or the reviewers at any point could say, this really isn't that good of a paper or doesn't belong in the journal. And so this process, the traditional process, can take months. So the paper that you see in a journal,
almost pure pain.
Of pure pain, exactly.
Pure pain. That's
of
it.
Almost pure pain. And there are internet memes about this. The dreaded third reviewer. So people talk about reviewer 1 loves your manuscript, reviewer 2 thinks you should have done their experiments for your paper, and reviewer 3 just hates it. It's clear reviewer 3 didn't understand the paper. I mean, of course, these are internet memes, and these have some basis in reality, but like Elio said, it's pure pain. So what mSphere Direct tries to do is to really put us as authors in the driver's seat. And so what I was able to do with this manuscript was to get rid of reviewer 3 and say, I'm going to pick reviewers who know my field, who understand why this is an important problem, and whose opinions I respect. And so I was able to write a manuscript and then give it to 2 people, Mick Watson, who's at the Roslin Institute in Scotland, and Catherine Pollard, who's she's at the Gladstone Institute in California. They're both well known for their skills in bioinformatics and for their research. And so they then wrote reviews of my manuscript. And the tone was very different. It became much more of a conversation between me and them. And so one of the reviewers had some comments or something they wanted to see, and I didn't really understand what they wanted. And in the traditional mode, well, I would try to answer it and then resubmit, and it might go back and forth until I understand them. And this time I generated what I thought they wanted, sent it, and they said, no, do this. And we went back a couple times, and that was over like 3 hours. So it was much faster. And at the end of all that, I got an email from Mick and from Catherine saying, we think this is a good paper and it's ready to go. So then what I did was to take my manuscript, my response to their reviews, biographical information about Mick and Catherine, to assemble all that, submit that to mSphere, and then that was handled by Trina McMahon, who was the editor, and then within 5 days, then the manuscript was accepted. And so, Again, the advantage to me is that I was able to pick the reviewers to be people that I respect and that I knew understood what we're trying to do or understand the importance. And so when we've described this to people, I think people cynically in the back of their head said, well, why didn't you just pick like Michele to review it? One of your colleagues, or pick Michael, and there are safeguards built into this that are pretty significant. So I had to pick people who had at least 5 papers in this area of research. These couldn't be postdocs. These couldn't be people
within the last 5 years, even.
Yeah, and I couldn't have published with them in the last 5 years. They couldn't ever have been my trainees. And ASM does a great job of having rigorous peer review, and so I know Mike Imperiale takes us very seriously. And I kind of also wanted to put on myself that I'm an associate professor with tenure. I don't want to give this to an assistant professor who doesn't have tenure, because I don't want them to feel like they have to suck up to me or give me a free pass. So I think I ended up giving it to 2 of the harder people I know. But I think it's a great system, again, to put authors in charge, to change the dynamic from, as Elio said, being pain to being more of a conversation. And anytime you can put it in terms of a conversation, it's going to be a more positive outcome.
It's also a much more transparent process because you obviously knew who was reviewing it. They knew that you would know, and their names are published on the cover page. And so they, of course, care about their reputation, so they're going to give it their most rigorous feedback. And then you also benefit because now when people read it, they know that it's had the careful, critical reading by 2 authorities in the field. So it adds like a good housekeeping seal of approval right on the front page. Mm-hmm. So I think it's a great system.
So Pat, was there any difficulty in getting people to do this? Because it's brand new, you might say. They could react like, what is this? Explain it to me.
Yeah, no, so it's funny because I'm also an editor at Applied and Environmental Microbiology. And so it's it was eerie how similar it was in some ways getting reviewers for applied environmental microbiology and for my own paper. And that I think I actually ended up inviting 5 or 6 people to review this. And like a normal manuscript, 3 or 4 of the people had to decline because they had too much going on. So, but I also picked Mick and Catherine because I knew that, at least in their public persona, that they're very committed to open science. And so they're people that I thought would get a kick out of this and find it to be an interesting model to participate in. So I think there might be concern going forward to see whether or not somebody would want their name on the manuscript. And they don't have a choice. So if that's a non-starter for them, then they can't be a reviewer, right? So it'll be interesting to see what happens. And one of the things I really appreciate about what Mike Imperiale is trying to do with this and other things at mSphere is that it's an experiment. You know, we're scientists. We shouldn't be afraid to experiment, collect data, reformulate our hypotheses, and modify things and go forward. So it'll be exciting to see what happens.
Well, I can only applaud this with all my heart. I think this does science at its best, where people communicate, interact, improve the situation, in this case, a manuscript. I don't think it could be anything better than this, and the risk is small. The risk being that sometimes something slips by which doesn't deserve to be published, but that happens no matter what. And I think the odds of that happening are very small. I really hope that the ASM and everybody else listens to this, looks at it, and takes it up in their own journals.
I was just going to volunteer that imagine if this had happened with that paper that was published in Science arguing that we had arsenic as in lieu of phosphorus in the nucleic acid, and the individuals who reviewed that paper had their names on that Science paper, then that was dismissed by everyone in the literature after they began to tear at the data. And you know, it's a footnote now in scientific literature that it's completely without merit. And I think this is going to be as rigorous, if not more so, because we've all reviewed papers, and if you have to attach your name to it and it's going to get published, you're going to make certain that when you say yes, it really is a yes.
Yep.
Not hope that somebody else fixes it.
Darn tootin'.
So, Pat, another novel component of this is the bioarchive link. Could you tell us more about that?
Yeah, so that actually builds off directly of what Michael was just talking about. So one of the things that we did and is also built into the submission process with mSphere Direct is to post your paper, your manuscript, as what's called a preprint. And a preprint is somewhat novel to the biology field. It's an interim research product. So when I wrote this manuscript, I wrote it and I submitted a copy to be posted on bioRxiv as a server that hosts these preprints. And at the same time, then I sent copies to Mick and Catherine to review. So Mick and Catherine could review it, but at the same time, anybody on this call or anyone listening to this podcast could have gone and got the paper and they could leave comments. So instead of having 2 people review it, I could have had the thousands of people listening to this as my reviewers. And so that does a couple things. So that expands the conversation. It also brings in perhaps more expertise. So instead of only having 2 reviewers who might have missed something, I now have the scientific community that can potentially listen to it. And so when you think about the Arsenic Life paper, if you can imagine that being submitted as a preprint at the time of submission, then when all the people who said this doesn't smell right read the preprint, that paper never would have even gotten published. And that paper is actually still part of the scientific literature. It was never retracted. And so the other thing then it does is that it gets the paper out there much faster. And so mSphere Direct is very fast in getting us a decision within 5 days. But as soon as it's accepted, it's marked as accepted on bioRxiv. And then it's hopefully going to be then published within a month on the ASM site. And so the tie-in with bioRxiv is great because, again, it expands the conversation and it helps the author then to disseminate the research that much faster. But then it also opens it up to that broader peer review and then speeds up the process of getting the information out.
And how does that work? I mean, is your first draft or the first draft of your manuscript that you submitted to the reviewers, is it still on bioRxiv, or that got replaced with your revised version?
Yeah, so it's not my first draft, but it's the draft that we submitted, right?
Right.
So there's different models. So sometimes, with that paper, there's a version 1 of the preprint that's still on bioRxiv, but then there's also a version 2 that was the version that responded to the comments of the reviewers. And so usually what my lab does is we'll submit to the bioRxiv at the same time we submit to the journal. ASM is nice because they have a little switch on the bioRxiv website that allows you to do a direct submission from bioRxiv. But we also had a paper about preprints in microbiology, of all things, that we submitted to bioRxiv, and I left it up there for 3 weeks to get people's feedback. And then based on that feedback, I revised the manuscript and then submitted it to a journal. So people can either do simultaneous submission, or they can post it, wait a little while, get feedback, incorporate that, and then submit it. It's an exciting time because there's a lot of different things going on with preprints and with publishing. And you may have heard in the last couple of weeks, the NIH has announced that for proposals and for biosketches and for progress reports that you're now going to be able to cite preprints. And so I think this will be very helpful to junior faculty who may not have a long track record in a field, or people coming into a new field where I might have 5 papers that are as preprints, but they're still in that long peer review process. And so to be able to point to that peer review or that preprint rather than some paper that's submitted, that's kind of what we put on our CVs and never quite know if that's submitted or if what's that. That's going to be an interesting game changer, a lot of us think, for publishing and for kind of moving people's careers.
So you have to have uploaded it to bioRxiv. In order to say it's a preprint, it has to be available on bioRxiv or a similar service.
Correct. Yeah, there's bioRxiv and there's also PureJ, which are bioRxiv is a non-profit that's hosted by Cold Spring Harbor, and PureJ preprints.
Hey, you said Cold Spring Harbor. Let me say something about just how great this is in a historical sense. Way back, when molecular biology got started after the war, the people participating in it all talked to each other, and they sent out preprints of their work. That is, when they had a manuscript in the making, as soon as it was anywhere near ready, they would send it out to all their friends. They use mimeograph machines to do that, which most listeners will know what that means. But in those days, the field was characterized by collegiality, by friendship, by a desire to move forward, and not by other things, not by motivations of trying to achieve and sort of be recognized. This, I think, this changed of necessity, partly because the field grew immensely, partly because of competition for money, etc. But this is a step towards those halcyon days. It's a step back towards having people focus on the importance of the science, on the necessity to share it, on the desirability of sharing it for the common good. So I really applaud this because it sounds just right. It sounds like the good old days coming back, at least to that extent. So I'm very happy to hear this.
So, Pat, how are you introducing your graduate students to this concept? Are you encouraging them to look at these preprint sites to see how seasoned professionals review papers, or what sorts of things are you doing to foster this behavior?
Yeah, so I try to give my trainees the freedom to do what they want. And so if they're still leery of preprints, I'm fine with that. I would rather that they post everything as a preprint and be as open as possible because, as Elio just said, I think we only have to gain by being open. I do encourage them to look at preprints, and when we trade around interesting looking papers, maybe 10% of the papers we share with each other are preprints. There's rapidly growing number of microbiology papers posted on bioRxiv. The other thing that I've had one graduate student who's finishing up really benefit from was that he applied for a postdoc, and he's in the process of that long, arduous battle with reviewer number 3. And he was able to tell a potential postdoc supervisor, hey, you can see my paper here on bioRxiv as a preprint. And that, to me, would, if I received that, that would tell me that this is more than someone's middle draft of a manuscript or some poorly formatted, paper, that this is like a legitimate piece of work if they are willing to put their name behind it and put it onto the preprint server. So at least that student is a believer, because it's going to help their career, because he's able to get a postdoc position faster and not have to wait for the reviewers to come back, and that postdoc advisor can see what he's done and evaluate it for themselves. I think that when I talk to people, they of course blame it on their PIs, that their PIs are being stodgy or conservative. And so I think this is a place where, like Elio said, if this is something that we believe in and it's going to help collegiality and move science forward, then we as now senior, middle career, senior career faculty need to be leaders and help kind of demonstrate that we're willing to review preprints and that we're willing to post papers as preprints to kind of help move this along and to get the maximal benefit out of it.
Pat, I have a procedural question. When you collect all these reviews, you go back and forth with your reviewers, and you get that. Now, what happens if one says reject, or they both say reject, and you can't get beyond that? Then that's the end of it?
Yeah, that's my understanding, is that that's the end of it then, that if the reviewers say reject, then you're done.
Okay. Now, what if it's one reject and one accept? You go to the third reviewer then?
That might be a better question for Mike, but I think if I'm the author, what I would do would be to maybe get a third reviewer and see if I can satisfy them. And the expectation in the mSphere direct submission process is that you give them the whole review history.
Right.
And ultimately, it's going to be an editor at mSphere that makes the decision. Mm-hmm. And so, I'm sure Trina McMahon, if I had 2 positive and 1 negative, and if she thought that the negative review was right, I'm sure she would have said, Pat, sorry, this isn't cut out for mSphere.
Yeah, right.
So I think, they're the final arbiter, and it's perhaps me trying to make a case as transparently as possible for why my paper should be in their journal.
Yeah. But the key here is that you get the paper reviewed, whereas at many journals right now, you can't even get it reviewed, right? And so you get the reviews and you put it in front of an editor, and they will at least look at it and consider it, and you're way ahead of Nature, Cell, and Science already, which won't even review your paper. So I like to say it lets scientists take back the publishing process in a way. You have a better chance.
Absolutely. Yeah.
I have one more question, Pat. On the title page, in addition to having the data publication, the editor, and now the 2 peer reviewers, I see the little Twitter icon. What's that about?
I think we put in a tweet. So one of the great things about things that mSphere Direct is doing, mSphere and mBio and other ASM journals, as well as bioRxiv, is that they're doing their best to help disseminate information about your paper. And so Twitter has been a great avenue. I know everybody on this call perhaps has a Twitter handle and are sharing papers. And so by clicking on the little birdie, the Twitter icon, it'll send out a tweet to my paper. And so that way then more eyeballs are looking at my paper and getting to learn about this material. And it's amazing how much, at least people in my lab, are finding papers because of people out there who will tweet other people's papers. So if you're not on Twitter, I'd really encourage you to check it out. And if you are, you should really be following people like Elizabeth Bick, who does a phenomenal job of aggregating papers. Cameron Thrash is another great resource for papers on Twitter. But it's again another one of those exciting things about publishing, where when I was a grad student, we would get the physical copy of Journal of Bacteriology and we'd leaf through the tables of contents every issue. And then as a postdoc, we'd get them emailed to us, and now they show up in my Twitter feed. So that way, when my wife yells at me for being on Twitter at home, I can say, I'm doing work here. I'm looking at the literature. She says, yeah, right. So
And other publishers are doing that too, and that's a skill set that I think we have to encourage. Is oftentimes, I know the Elsevier family of journals is asking you to condense your abstract to graphical abstract, because again, they're looking for easy and convenient ways to publish size and drive traffic to their journal titles. But the beauty of mSphere is it's in the open literature. It's in the public domain. It's not behind a paywall, correct?
Right.
Correct.
Pat, do you think this is going to spread to other journals at some point?
I think it is. I heard anecdotally from Mike Imperiale that, I forget who the editor was, and it doesn't really matter, but that they were frustrated that we came up with the idea first, because in hindsight to them, that seemed so obvious. But so I think other journals are going to adopt this as well. And again, we need other people to do it. That my co-author Sarah Westcott and I were the first people to get through this process, and we need other people to do it too. That if it's something people believe in, then they need to follow along. And I'm a big believer in leading by example. And so if we think this type of communication, openness, and being able to drive the process as scientists is important, then we need to do it ourselves to show those younger generations and our colleagues that this is a viable method.
This episode of TWiM is brought to you by Blue Apron. Blue Apron's mission is to make incredible home cooking accessible to everyone while supporting a more sustainable food system, setting high standards for ingredients, and building a community of home chefs. What they do is they deliver seasonal recipes together with fresh, high-quality ingredients so you can make great home-cooked meals. You get a blue box. I'm seeing them everywhere now. I drive down the street and I say, there's a Blue Apron box. Comes to your house. It's got in it a step-by-step recipe card, gorgeous card, which you can save and do again if you'd like, because they don't repeat their recipes in the year. So it would be another year before you got that same recipe. So if you love it, you can go out and buy stuff and do it. And they give you all the pre-portioned ingredients you need to make a meal in 40 minutes or less. And it's going to be pretty inexpensive, less than $10 a person. So if you eat out and spend a lot of money, or if you go to fancy grocery stores and it costs a lot, this is going to be very economical. How do they do this? Well, they have a network of farms and fisheries and ranchers across the U.S. And then, of course, they have warehouses where they ship their food to, and it gets all put together in boxes. And I know this because they're building one in a town near me, a new warehouse. And all of this is very fresh, and it comes from places where they do responsible farming. For example, they get seafood from sustainable sources. They get beef, chicken, and pork from responsibly raised animals, produce from farms that practice regenerative farming, and they give you just exactly what you need. They give you everything you need except salt, pepper, and oil, but it's exactly what you need. So if you need soy sauce, if you need 2 teaspoons, they give you this tiny, cute bottle of soy sauce, which is all you need. So there's no waste. Plus, you don't overeat. You can order for 2 or 4 people. I guess if you ordered the 2 and you ate it all yourself, you'd be overeating, but you're supposed to share it with other people. So you can customize your recipes every week. You can get meat or non-meat, of course, and you don't have to order every week, whatever schedule you want. And they deliver to 99% of the continental U.S. No commitments, and you never get bored because they don't repeat their recipes. Here's some of April's recipes. Spinach and fresh mozzarella pizza with olives, bell peppers, and ricotta salata. Sweet and sour salmon with bok choy, carrot, and ginger fried rice. Parmesan crusted chicken with creamy fettuccine and roasted broccoli. Wow. You should go to the website and see all the other recipes they have. Check out this week's menu. You'll get 3 meals free with your first purchase, with free shipping on top of it. Go to blueapron.com/TWiM. That's blueapron.com/TWiM. You'll love how good it feels and tastes to create incredible home-cooked meals with Blue Apron. So don't wait. Blue Apron is a better way to cook. So, Pat, what about the science in this paper? Can you tell us what it is about?
Yeah. So, it's I wouldn't say it's as exciting as 95% of the papers that you all review here on TWiM, but it's a really important problem. And so pretty much going back to that curvy thing that, that plot, the refraction curve that Joe wanted when I was a postdoc, the fundamental problem was, how do I take all those sequences and put them into different clusters based on similarity to each other. And so one of the dark secrets that your listeners may not appreciate is that we don't know what all of microbial life looks like, right? Like that's hopefully obvious, but we haven't seen everything. And I have a family member that will call me and says, have you discovered any new bacteria recently? And I say every day that I can go into anyone's gut and I'll find bacteria that we've never seen before. And so that's one of the things I love about microbiology, but it's also very frustrating because that means that our databases are limited, that I can't take an unknown sequence and find a perfect match for it in a database. And so what we do instead is we try to clump sequences together based on their similarity to each other. And this is what we call an operational taxonomic unit. Some people kind of colloquially might call that a species, but that's a whole hornet's nest that you don't want to get into. So this, as you mentioned in kind of the intro to all this, generating all this data requires new tools, better use of our computer hardware. That the tool I developed with Joe was called Daughter. And if we'd have had another Daughter the year I wrote Mother, we would have been Daughter 2.0, but instead we wrote Mother. And so Mother then was kind of the next generation. And the algorithms there were faster and better and did a better job of handling these big data sets. But the data sets keep getting bigger. And so that forced us to develop this new algorithm called OptiClust that's faster. It uses less RAM, less memory, and it's actually better than any of the other methods that are out there. It's a really important part of the process that we use when we analyze 16S data. So whenever you hear about a microbiome study, those microbiome studies all depend on tools like this to take those millions of sequences and to tell a convincing story, an interesting story, right? So it goes back to that problem again of, I have a postdoc who's looking for the role of the microbiome in colon cancer. We've sampled 500 or 1,000 people, and we have thousands of sequences from each person. How can I process that? My computer's going to go up in smoke. And so we developed this new algorithm to make that optimized for quality of the clustering as well as speed and memory utilization.
And the first author here, Sarah, she did most of the heavy lifting?
Yeah, so Sarah Westcott is a programmer who works with me. And Sarah actually worked for me while I was a professor at the University of Massachusetts, and she actually still lives in Massachusetts. And so we Skype frequently, and she came out to visit Ann Arbor last year, and I said, I've got this problem, this idea I've been kicking around. And she said, I could do that. And so if you've ever used Mother and you've ever emailed us for help, you've probably gotten an email back from Sarah. And Sarah's just a really bright, creative person who does just amazing work and made this possible and kind of took my scattered ideas and what I was writing up on a whiteboard and put it to code. And what we've heard from users so far is we've got a lot more very happy users.
So this is really interesting. You don't even have to be in your lab to do the computational biology, right? Because there's no wet work and you could do it anywhere in the world, right?
Yeah. I mean, we do have a big wet component, but this is a dry component. So it's worked out well. And of course, that's the beauty of, we're all able to talk to each other today because even though we're not in the same room, right? It's the same kind of thing.
Yeah, absolutely.
What does OptiClust mean? I can tell the clust means cluster, but what is Opti here?
Yeah, so it's short for optimize. So
optimize.
We have
Thank you.
Yes. Yep.
And the paper is full of quantitative data, by the way, showing that how their method compares to others in the field.
Mm-hmm.
Yeah. And back to the thing about Sarah not working here in Ann Arbor, that I think it's been really good for her in terms of work-life balance, and that she's told me that she's far more productive working from home than she was actually when she worked in the lab, which is interesting. But so it's a great project, and it's been a great collaboration that we've had over the last 10 years or so.
Yeah, I could be more productive without a 3-hour commute too. Can't do much in the car except listen to podcasts. So Pat, you're using this in your lab, and you expect others to be adopting it as well?
Yeah. So if you're a Mother user at this point and you've got the latest version of Mother, this is now the default algorithm that we're using for clustering.
Mm-hmm.
And so, so it's out there. It's in our software. Like I said, it's not as fancy as some of the papers that you see on the microbiome and all the exciting things that are out there. But as our data sets get bigger, it's going to be critical to making the types of inferences that we want to make about the microbiome.
And so obviously people can get this online, right? The code for this.
Yep. The code is all open source, which means that you can get it for free. You can look at the code. You can say, hey, Pat and Sarah, you could have programmed this better, and you can give us new code if you want, or you can take it and use it like everyone else. The website is mother, MOTHUR.org, and we've got a pretty well-developed website there with a lot of documentation to learn more about how to use the software.
To pick up on something that Mike just said a while ago, what kind of training do you think one should give young students, entering students, in order to be able to use tools like this? How should one modify the programs, the teaching programs that exist?
I think we need to do a better job of integrating quantitative questions into our normal classes. I remember doing things like, Michaelis-Menten kinetics and Monod equations. And of course, I came from an engineering background, so that was comfortable to me. I remember teaching when I was at UMass a microbial ecology class where I had them solve a differential equation, which was like dx dt equals mu, which is differential equation for exponential growth. And a prerequisite for our program was calculus. And the students were not happy. And I said,
they're still not happy. I tried to do that, and they get really militant.
Thomas and Finney's calculus book is over in the stacks. Go get a copy of it and figure it out. And that's perhaps a silly example, but
is that sufficient? Is calculus sufficient?
No, I mean but I think probably more important than taking calculus is seeing these examples embedded in the classes they're doing now, right? So my poor 13-year-old is trying to figure out how to factor polynomials and doesn't understand why it matters. But if that was in biology or if that was in physics where he had to see why that mattered, then he'd be going after it like gangbusters, right? So, and if you think about, here's this growth curve data from E. coli growing on different types of media, how do we calculate mu max or the Km or, these different parameters? And then also thinking about just the troves of sequence data that are available to us for free that probably largely go underutilized, that you could go into GenBank and you can get thousands of genomes at this point and do comparisons. And so, but our students don't have that ability. And I think if we have microbiology students leaving a program in the era of genomics and metagenomics and they don't know how to work with genomic data, they're going to be hurting.
That's exactly the point. I think they're going to be out of a job. But you see it from the point of view of being an engineer, having all the quantitation you need under your belt. But I'm not so sure that it's enough to say, let's include this in our classes so that the students can see why it's so important. I mean, it has to go back a little bit further back, I think, to high school maybe, and get a better quantitative preparation all around.
Absolutely.
You can't walk into a class in biology and have to solve a differential equation and be done with it. That's not enough.
No, right, and it's they've got to have the raw skills. That one of the things that I've been involved with and I've brought to the University of Michigan is a group called Software Carpentry, and they have a sibling organization called Data Carpentry. And actually, Michele took the Software Carpentry workshop last time I took it, and that's to teach people a lot of the initial skills you need to be comfortable at the command line, because all these tools that we use that are really powerful are at the command line, and how to use software like R, and how to, keep track of our data, and how do you keep a computational notebook, because it's different than a wet lab notebook, and these types of things. And I think if people see numbers around them and think about how can I use these numbers, which are really describing the biology, to answer interesting questions, then I would hope that that would make it more useful, right? So again, going back to my poor son learning algebra, if he saw why those things matter in the context of the world around him, then he'd love math more, right? At least I hope he would. But if that was baked into the biology, it's kind of like we have this problem in biology where do we teach students this is how you dissect a frog, or do you give them a question to answer by dissecting a frog, right? So much of it's what Joe Handelsman used to call cookbook science, right? Where you just kind of follow a procedure and you get, quote unquote, the right answer rather than being experimental based. And so I think there is, at least from my perspective, we need a lot of change, both in terms of being more experimental in how we're teaching students in biology classes, but then also bringing to that numbers and quantification and not being afraid of that type of content.
Michele, you a coder?
No, but
you took a course.
We are anticipating doing more genome analysis, and so I wanted to begin to get those skills.
Maybe you'll be using OptiClust, huh?
I think I will. It'll be baked in, right?
That's right. Well, I like the sound of it extremely so, but I in the past, it used to be that one had to ask students to come better prepared. I remember in my old department in Tufts, we demanded that graduate students entering graduate students come with a course in physical chemistry, and if they hadn't had it in college, they had to take it in the summer before starting their work. And I was always surprised how often students felt that this was a good idea. Now, maybe this is not required anymore, but sometimes a little bit of quantitation, maybe force-fed quantitation may not be a bad thing. I don't know.
One of the things that the branches have been doing, especially the Florida branch.
We're talking about branches of the ASM.
Branches of the ASM is what they have been doing is they have been appreciating what Elio is talking about. And so branches traditionally have undergraduates, graduate students, postdocs, and faculty. And they've been running workshops on basics of R, advanced R, and other things. And we have been sponsoring those activities out of the branch organization committee because I've been listening to Elio a long time and I've picked up some of his wisdom and thinking about how the next generation and, these courses out there with an applied bent towards things that we need to get done in the lab aren't being offered in a traditional undergraduate program. And Chris Kellogg, who was an at-large counselor on the old ASM governance structure, she was the strongest advocate. And Chris works in this field working on marine microbes. And of course, if Pat finds a new microbe out of the colon every day, she at least finds 10 out of seawater because that's truly the undiscovered country out there. And so I think we really need to be clever to offer these opportunities to students of all ages so that they can become adept at it.
Absolutely. And I also don't want your listeners to leave this feeling dejected because they don't have those skills. That the truth is that I've probably had 6 postdocs work for me, and I've had 5 or 6 graduate students. And I think one of them has come into my lab with the type of computational skills that I would hope for them by the time they leave my lab. Very few of them come with any programming expertise. And so one of the things I tell prospective students when they come to do a rotation or for interview weekends is I say, if you're to get a PhD in, say, history or philosophy, before you could have your qualifying exam, you would need to be proficient in several foreign languages, say French, Greek, German. In my lab, we have a language requirement too. And it's not that you learn a spoken language, but you're going to learn R. And we're going to help you out along the way. But really what's going to happen is you're going to get 10 million 16S sequences. And I'm going to do that thing Joe did to me. I'm going to say, hey, can you make this plot for me by the end of your rotation? And we do a lot of support to help people along. But you got to think of it as a foreign language. And
that's beautiful. I love that analogy.
I appreciate that people aren't going to come to my lab with those skills. And frankly, I worry that they don't come to me because they don't have those skills. That we're happy to train people and work with people. It gives me a lot of joy to see people get it and to grow in those skills. And at the same time, learning great microbiology and helping to advance the field.
Well, that is great.
Here.
I love it. I'm lining up to learn R.
Great.
My last name begins with it. I have to learn.
There are a few more sequences out there to compare your polio genome to now, huh?
Well, that's a good question.
You could probably do it for Zika, Vincent.
Well, nowadays people do lots of viral sequence comparisons because we have the algorithms to do it, but we didn't have them back when I was a postdoc because there was no need for them. You couldn't sequence enough. Remember, it took me 1 year to do 7.4 kb.
Right. But now you can ask all new questions.
Totally new questions. And that's why we have an influx of computational biologists into the field. And, one of the things that I like to say, Pat, is they come in and they really don't know a lot of biology, and it's our job to teach it to them so that they know where to apply their skills.
Absolutely. Yeah. If they came to us and they had all the skills, then what's our job?
Yeah.
We've got, and if they came in and knew everything, then we should give them their degrees on opening day, but that's not the way it works.
Well, this has been a great conversation, and I'm going to make a editorial decision that we end the episode right here, and we'll save the paper that we had until next time. Is that okay with everyone?
It is.
Perfect.
Oh, yeah.
Because I think it's self-standing.
This is a departure from our usual, but boy, what a nice departure.
Yes, it is. That's
Pat,
TWiM.
thank you so much. This is really great.
My pleasure. Thank you all.
This is TWiM 149. You can find it at iTunes, microbe.tv/TWiM, and also asm.org/TWiM. Consider becoming a patron of TWiM and all the other Microbe TV science shows like TWiV and TWIP. You can go over to microbe.tv/contribute and find the ways you can help us, Patreon, PayPal, and much more. And we love getting your questions and comments. Send them to TWiM at microbe.tv. Michele Swanson is at the University of Michigan. Thank you, Michele.
Thank you.
Elio Schaechter at the wonderful blog Small Things Considered. Thank you, Elio.
Thank you. How nice of you. Thank you.
Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thanks, Vincent. Thanks, Pat, and everyone else.
Our guest today at the University of Michigan, Pat Schloss. Thanks so much, Pat.
My pleasure. It's been great to be with you all.
Tell us your Twitter handle so everyone can follow you.
Yeah, it's Pat Schloss. One word.
Straightforward. I like that. Thank you. I'd like to thank the American Society for Microbiology for their support of TWiM, Chris Condayan and Ray Ortega for their technical help. I also want to thank the sponsor of this episode, Blue Apron. The music you hear on TWiM is composed and performed by Ronald Jenkins. You can find his work at ronaldjenkins.com. Thanks for listening, everyone. See you next time on This Week in Microbiology.